Good morning, good afternoon, good evening, depending on where everyone is in the world. My name is Kate Marshall. I'm Technical Director at Protac Scientific, and our company focuses on hydrogen peroxide bio-decontamination validation, hence the presentation today. Just before I start on to the presentation, just remind everyone that the session is recorded, so you'll be able to actually watch the webinar back. And also, if there's any questions, if you could pop them into the chat box. I have a question and answer session at the end, so we can run through those rather than having questions throughout. So we should get started, hopefully. Okay, so in terms of agenda for today, what we're going to be looking at is bio-decontamination validation with hydrogen peroxide, and so that you can learn a little bit more about the enzyme indicator technology that Protac manufacture and how it's helping the industry understand bio-decontamination. So in terms of really setting that scene, we'll start looking at what the Annex 1 requirements are now. What is hydrogen peroxide bio-decontamination used for? What the process of validation is and how it's impacted throughout these stages, how the traditional technology is used, and how enzyme indicators are a benefit for this process, as well as some real data that's been gathered across the industry, particularly from our pharma working group. And then a short case study that we have from the NHS as well in this field. So just to give a brief background on myself and why I'm here today. So I started my career 25 years ago in pharma and worked in several roles through GSK, focusing on aseptic manufacturing. So I started to specialise into different areas, a lot of time spent in validation, which is where I started to learn about bio-decontamination, was subject matter expert for some of the projects and the equipment that we were installing, and started to find some of the challenges and pitfalls as we went through that process. And towards the end of my career at GSK, I found out about the technology, realised how much it would benefit the industry, and saw an opportunity to join the company. So I've been with Protac Scientific for 5 years now, and I now head up the technical side of the business. So that will be from supporting our customers with use, helping them look at their validation strategies, how they can be used and implemented in the process, as well as what we do internally with our product. And a brief overview of Protac. So we were founded in 2014 off of research by Public Health in England, which is now the UK HSA. We're working with over 50% of the top 20 pharma companies, and this is from starting to use the technology through to global rollout of the technology for all their bio-decontamination processes. We're very strategic as well, with really trying to focus on working with the equipment manufacturers to really get those robust, repeatable cycles and fit-for-purpose validation for the equipment that's in use for really critical processes around the world. And webinars like this through CCN and also through other representation and using case studies for customers is really about how we're showing how the enzyme indicator is being used in the industry. So if we start with Annex 1 and bio-decontamination, the latest revision of Annex 1 that came out and was live last year, but now is in place since August, there's been some updates that really focus on bio-decontamination. And when you start to look at some of the information, this is some of the highlights of that information through Annex 1, you start to see areas where, from a regulatory perspective, they're seeing challenges to the validation and calling out in specific areas that need to really show that that process works. Now, a lot of this is focused on isolators. However, clean rooms, that control and that validation still is important. So these areas will then focus into those, just because it's not necessarily called out in that way. If you think about RABs in a clean room, the goal is to almost have the clean room decontaminated as well as that RAB. And we see that set up sometimes. So the applicability of these requirements really fits any purpose of bio-decontamination. But this understood and validated, so 4.36, use of hydrogen peroxide, what the effectiveness is, understanding that effectiveness, and then also that it's understood and validated. And it's fit for purpose, which is the goal of the validation on any validation that's being executed. So in terms of that bio-decontamination life cycle, what is the hydrogen peroxide used for, or bio-decontamination used for? So traditionally, it was vaporized hydrogen peroxide that was used. We're seeing more and more different application methods, but it's a substance that destroys or eliminates all forms of microbial life in the inanimate environment, including all forms of vegetative bacterial spores, fungal spores, and viruses. So a sporicidal agent that's being automatically put into an enclosure space. Now, that enclosure could be, traditionally, it was all sterility testing isolators. We're seeing more and more complex manufacturing isolators, pass-through chambers into clean rooms, or the clean room itself. So lots of different applications, but the fundamental part of the validation and the steps will remain the same. So regardless of whether this depicts an isolator, transfer chamber, the process starts with a hydrogen peroxide generator, and the hydrogen peroxide is put into the enclosure space. So once it's put in, it tends to be a conditioning phase. There's an amount of peroxide that's put in that's higher for a short amount of time. It will then go into a holding phase. So this tends to be a decontamination phase, where less is put in, but a longer duration. And then it's an aeration where the hydrogen peroxide is removed from the enclosure space. When it's a clean room, this is a bit more complex because it's relying on the HVAC of the room rather than a set fixed isolator setup of HVAC. So that extracting of the peroxide is what takes the majority of the cycle time. So the amount of hydrogen peroxide that's going into that enclosure space is really critical and key. How is it validated? This is where samples would be put. So indicators are put around the enclosure space. This could be chemical, this could be biological, and now the enzyme indicators to look at challenge locations and really trying to understand what areas are critical to that process and worst case locations to then gather samples. So those samples are put out. The process is run through in this standard way that's been set up. So you have the conditioning, you'll have the decontamination, and then you'll have the aeration phase. And then the samples are removed and tested. So as a run through of a process, put samples out, run the gassing, collecting, it looks overall very simple. But what's the technology and what are the challenges with that validation? So if we look at the process of that validation and the steps that you would go through to prove that that cycle is repeatable and robust and validated, that starting point of the engineering cycle. So again, applicable to a clean room, applicable to an isolator, understanding how mechanically that's set up. Those initial cycles are showing that the system is capable of achieving a 6-log reduction of biological spores. Intercycle development, where you're looking about how that's going to be used in routine use on a worst case requirements. And then once you've set a cycle, the performance qualification should be a repeatability. So you go from your setup of is it a robust cycle into is it repeatable and performance qualification? And does it repeatably show a 6-log reduction? Now, once the performance qualification is complete, the system would be in use. And then the periodic requalification comes into play. So that could be 6 to 12 months' time, where you're then doing a repeat of the PQ parameters, normally one cycle, to look to make sure that it's still performing. traditional technology that was used was biological indicators and still is a requirement of some levels. If you notice on the Annex 1 update, it doesn't talk about biological indicators anymore. It talks about a bacterial reduction, and that's applicable for the system. Now, biological indicators make that easier to have something with a known challenge level to put out in the enclosure space. So the process in which they're used is the biological indicators are put into that process. Once that process has been run through, they're collected and put into a growth media and then incubated. Now, after 7 days of incubation, that's the final result as a minimum. Some people may incubate them for longer. There's some requirements that's 14 days. What that gives you is a pass-fail growth, no growth result. So some of the challenges with biological indicators is making sure that it's representative of the challenge that's needed for the system. There's lots of reports of clumping, which means that if they're not in a nice monolayer, the spores aren't behaving, then you could have biological indicators that are still showing a growth when actually it's down to the setup of the biological indicators themselves. Also the approach, most probable number, so having to use triplicate to then protect against if there will be any failures, means that you can have a large amount having to be used in the cycle development and validation. So if you take the biological indicator challenge itself, and then what else can actually affect that efficacy of the peroxide going into the chamber and enclosure space, you can have temperature impact of the peroxide going into the space, and that can be a differential in temperature. So the temperature at which it's being generated versus the temperature control of the enclosure space. So for an isolator, this might be able to be controlled quite tightly within plus or minus 5 degrees. For a room, there is an element of operator comfort, what that set points are, and how that room's controlled, especially if you're retrofitting a gassing process onto a clean room that's been set up and is robust enough for clean room operations. So that differential is important, because if the peroxide goes into the chamber and the temperature is too great of a difference, you can actually lose the efficacy of the peroxide as it goes through. And that means you have a variable result. So how do you have that repeatable, robust result? Humidity as well. So this plays a huge part. And that tends to vary quite a lot, and can vary quite a lot even on isolators. So again, starting point of humidity, and it will be different across the enclosure space. So even the smallest of isolators or rooms could have quite a differential in humidity across the space. The quantity of peroxide, so making sure that it's really clear how much is being used and that it's precise. It's really down to how much is being injected in, rather than actually using a ppm measurement, parts per million of peroxide. That gives a good indication if you've got a gross failure of the efficacy. So if you start seeing low measurements of the peroxide concentration, you can start to understand if there's been any impact. However, it's a byproduct that can fluctuate quite a lot and doesn't necessarily mean that you're seeing consistent results across the whole chamber. Couple the load that you're using as well, so the impact of the items that you're putting into that enclosure space. So just as applicable to a clean room, what items have you got in there? Is there trolleys? Is there materials? Is there items that may be permeable to hydrogen peroxide, which is where this material of constructions become important. So you'll have different results based off of hard plastics, stainless steel, glass, as you would do to permeable materials. And then also the biological indicators themselves. So all of these factors can affect that efficacy and the repeatability and robustness of the cycle. And it's not normally very clear if it's one thing that can cause it. So having a really good investigative approach and being able to look through that data is really important and key to the successfulness of the gassing. So if we couple all of that together and what those challenges mean through this different life cycle stages, looking at the BIs that are going to be used, does the system actually set up and is it representative of how it's going to be used? And is it optimized mechanically? So how do you know that the set points, the amount of peroxide going in, how much is being removed from the system? Are there any leaks? That's all really important to its repeatability. How many biological indicators or challenge indicators to use? What's the approach to the gassing? So do you perform the validation on a half cycle and then double the amount of peroxide or time to actually achieve a predicted log 12 reduction? That approach means that you might not have actually gained anything on the efficacy, but you've doubled your gassing time. And that long injection amount of peroxide going in will then double, if not quadruple, the amount of aeration that's needed to then bring that down to safe levels for use. And periodic requalification. What happens if 6 to 12 months later, you then have a BI failure? You're then having to assess everything that's been done in between the initial validation to then the requalification. So where is it that the enzyme indicators bring value to all of the parts that we've just discussed? So if I start with a similar schematic here to how the enzyme indicators are used, they're small, flexible pieces of plastic that have a mesh on the bottom that contain the enzyme, which is thermostable adenylate kinase. In terms of their size, one biological indicator wrapped in Tyvek could house 3 enzyme indicators. So in terms of size and being able to fit into lots of different areas, there's a good flexibility around their use. Where this then differs, they would be exposed to the process, they can be taped, they can be marked up in a similar way to biological indicators. They're not put into a broth and incubated, but actually run through a bioluminescent assay controlled by a reader that has a photometer. So we're collecting the levels of light that's been produced at the end of the process. And that's all captured on a digital software. Now, the biggest difference in time and the time it takes to get a result is that each indicator takes 60 seconds to achieve that result. So the time in which you're running those samples is dependent on how many you have in an enclosure space. But that's a significant time saving compared to having to wait for a 7-day result. In terms of robustness, repeatability of the enzyme indicators themselves, it's a very different manufacturing process. So it's more of a chemical process for the enzyme itself, which means that there's a lot more control in that manufacturing. So biological indicator variation can be -50 to 300% variation. And that's an acceptable range based off of what the manufacturer has claimed. For the enzyme indicators, it has to be less than 15% variation for the batch to be put into commercial use. And the data you get from one single EI is quantifiable. And this just explains a little bit more about how the process works. So if we consider that this box represents what happens inside the reader, the enzyme indicator is placed into the reader, which contains the thermostable adenylate kinase. And adenylate kinase as an enzyme is present in all living cells, and it's responsible for energy management within a cell. So the reason for the thermostable aspect of this enzyme is it's been taken from a thermophilic bacteria. And it's got this hardiness to temperature, humidity, and robustness to actually give a measurement and show an inactivation profile. So once it's put into the reader, there's 2 reagents that are used. The first one contains luciferin luciferase, which is the known combination of enzyme and chemical reaction to produce light. So we use that to marker the enzyme that is left on the strip at the end of the process. But what we're looking for is an amplified reading because the enzyme itself will catalyze ATP. So to do that, we inject ADP in, And then we actually have an elevated reading of light as the enzyme converts the ADP to ATP. So we have a sort of joint use of luciferase and the thermostable adenylate kinase to produce light. And this is all captured in the software, and the photometer takes a snapshot view that gives a relative light unit reading. So if we see a high relative light unit reading, it means that we've actually got a lot of enzyme left on the strip. So the process hasn't been very good at inactivating the enzyme. If we see low levels, that means that we've got less TAC on the strip, and we've actually got high process efficacy because it's inactivated that enzyme. So you get a measurement and a scalable result of the efficacy. And how that all came to be and how the company was formed was off the research by Public Health England, and actually doing this correlation study between EI and BI results. And that's where this biphasic inactivation profile and similarities was discovered. So what the software allows to do and how this works is the samples were taken of EIs and BIs at different time points through a gassing cycle, which gives a correlation curve and a data set that means that you can predict what log reduction of biological indicators has occurred through your cycle. So this would have been a snapshot view from one cycle on one setup, but it gives you a gauge of how efficacious that cycle is, and if you're expecting to see biological indicator growth at the end of that. So it speeds that whole process up of being able to make really good data-driven decisions of where to go with the validation and what may impact it. And to put that into sort of real-life use, this is some data that's been gathered by our focus group from Pfizer. This data set shows how they've used it to look at material permeability, compatibility with the process, and also load configuration set out. So this actually shows the relative light units from different EI samples put across 49 locations in different configurations. And you can see that there's a level of inactivation that's been shown, and areas where they're slightly higher. When we looked into this further, those higher readings coincided with permeable materials. So it's about how much peroxide is being absorbed into the packaging and can lead to variation of results. Now that then in turn means that if a biological indicator is on that surface, this could mean a positive or a negative BI result. And it's the variation that you want to start to look at to control. So they could use that to then look at what configuration gave them the best outcome. So if we speak a bit more about that challenge of materials, and when we feed that back into the Annex 1 requirements, where it talks about gloves and what the impact of gloves and making sure they're well characterized with challenge locations, this data really shows that point. So this was a study with Optima, Metal and Plastics Group, and looking at different materials and what those readings would look like from the enzyme indicators from a permeability perspective. So you can see the graph here where we have glass and stainless steel, which have similar inactivation profiles for the enzyme indicators, with gloves being a lot higher. Now that again represents that we haven't managed to reduce that to similar levels that you would see on a glass or stainless steel surface, and more variability. So that canvassing of gloves is really important. And this has been studied extensively by Aerex, who are an isolator manufacturer, and really trying to understand how computational fluid dynamics and modeling of gas flow can then be used with real-time data off the enzyme indicators. What they saw from their study and what they were concerned about was the low levels of gas flow underneath the bottom surface of the glove, and what did that mean to their results. So they could then use the enzyme indicators to map the whole glove and look if they was to see if they were having similar inactivation profiles or if there are any problem areas. Which again feeds back into Annex 1 requirements about really having them fully understood of the surfaces. What they found was actually the bottom surface of the glove had a better inactivation profile than the top surfaces. And that comes down to the disruption and distribution of the peroxide as it comes in and hits that surface. So although the CFD showed that that area may have low flow, it was a lot more consistent area of gas concentration. In terms of optimizing cycles, this data from AstraZeneca really shows that benefit of what the data sets can bring to have a robust, repeatable and optimized cycle. And this study in particular was to look at how long the cycle was, how much gas was being produced and put into the system. And these bars actually show the log reduction potential against biological indicators. The study was performed over 3 days, and the log reduction, the yellow and the green bars here, represent their initial cycle versus an optimized cycle that was all confirmed once the work was completed with enzyme indicators using biological indicators. The difference between the yellow and the green bars here are 4 hours. So the reduction in the amount of peroxide actually led to a similar inactivation profile. So the log reduction potential of the cycle was similar across, but there was 4 hours difference. And that was because we were able to actually reduce the amount of peroxide that went into the system, which in turn reduced the amount of aeration that was required to remove it. So that meant they could look at how much production, how much testing they could do on those isolators, and actually then look at their throughput and achieve more throughput on that usage. If we think about validation delivery times, so the time it takes to actually wait for those samples to come off from biological indicators, a lot of running through the validation at risk with initial results. Again, that comparison of enzyme indicators being used in a study from Roche here, 6 test cycles were conducted in 3 weeks to allow them to get into qualification versus what would have equated to 17 days of 17 test cycles over 12 weeks and then into qualification. So the data and the decisions enabled that to be a lot quicker to move forward and have a robust cycle. And this example was actually on a full clean room and RAB setup. So you can see some of the data that the Pharma Working Group has got together, and the goal of this group has really been to look into how they can be used and compare the way they're being used to start to get some standardization. One thing that's really key in the industry is there's very different strategies across different companies. There's very different ways of looking at the guidance, in particular how Annex 1 is being viewed and looked at for lots of different subject areas as well, and interpreted, means that you can end up having different approaches. And we were fortunate enough that members from these companies wanted to get together and actually share how they were using the enzyme indicators and really push that forward within the industry. And we meet on quite a regular basis. We have representatives, a couple of representatives from each company, and meet on a fortnightly basis to discuss how they're being used, how we can start to push that information into industry and start to benchmark and standardize their use, including this routine process verification use of the enzyme indicators. And all of the discussions that we've had have all started on all of the topics that we've just covered. So BI strategy, do you use 1, do you use 3? What D value do you use? What are the challenges of the validation? What's that regulatory approach to how to use enzyme indicators and how do you use the information? And what are the strategies that are in place? And that really highlighted, I think, to everyone that joined initially, how much variation there is in the industry and how important it is to have a holistic combined approach. So one of the real goals of how that process of validation would work with enzyme indicators is going back through these phases. So really looking at how they can be optimized mechanically, how the system can be set up, and really be assured at that stage that the system is capable of delivering for the end use. And really putting more effort into the front stages to make sure it's mechanically set up. The data that you get from enzyme indicators can be analysed against all different parameters to really understand if that setup is right, rather than a calculation that maybe I have this enclosure size, I'm going to put this much, I'm going to calculate how much peroxide goes in. This really gives you that feedback tool to understand if that's correct. Then into cycle development, performance qualification, use of biological indicators to begin with still in those phases, but you can have a much more targeted approach. So you can use the most probable number approach, but maybe less positions because you've truly understood what your challenge locations are. Canvassing and having a guess and having a rationale that's based on understanding what the risk points might be. You can use the enzyme indicator feedback of data to actually ratify and reduce those positions, and then have a real targeted approach with the biological indicators, and have an optimized cycle that gives you more than a 6-log reduction to prevent the change of d-value of biological indicators from having an impact on that cycle in the future, which then means your aeration isn't lengthy, and if you then have to add anything onto a production cycle, it doesn't cause you any operational issues. And the periodic requalification would still feature. However, process monitoring is possible with enzyme indicators. So you could do a periodic check of the efficacy of that cycle, ensuring that it's still robust, repeatable between your periodic requalification and your from your initial validation. So you have an early alert system there that would tell you if you have any problems with your cycle before you've committed anything to the operation of the room or product to the isolator, and reducing that risk of then having to perform lengthy investigations and put product at risk. So a bit of a whistle-stop tour through that process, but this really puts it into perspective as to how they have been delivered in a project. And we're fortunate enough that because it was with the NHS, we can share this information and talk about how they've really given benefit to the NHS in an area where they're really starting out quite new into the use of bio-decontamination with hydrogen peroxide and what that validation means. So enzyme indicators were actually used across 8 isolators on Liverpool Hospital to really set that goal of being able to understand the efficacy and impact on aseptic compounding isolators and the criticality of that. So compounding products that are going straight to patients for use, and that criticality that they remain aseptic and sterile. So this approach was a full life cycle approach. So there was a lot of other work that Protac Scientific formed. We looked at load configurations, we looked at racking, we looked at how those isolators would be used, and really started to challenge how they could be used for routine use to make it easy for operators to use the isolators and to make sure that the throughput was okay for the cycle times. Looking at distribution and challenge locations, limit testing, really understanding that through the cycle development. What are the boundaries of a robust, repeatable cycle, and making sure that we did that with enzyme indicators as well as biological indicators. And then once they're into that routine use, so the performance qualification, and then the goal is for this continuous monitoring. And they're just at these stages now, past performance qualification and starting to implement the continuous monitoring. So very, very complex setup of isolators, lots of consumables and products that were required in the isolator. So a big mix of items that had to be assessed for what their compatibility was with hydrogen peroxide, Were they permeable? Did they have any risk to personnel or product exposure? And coming up with a racking that helped them have a mixed loading approach and flexibility around how they would be used. Where the enzyme indicators really gave value on this product was being able to map 8 isolators and understand where our at-risk isolator was, that we could do a lot more work and a lot more cycle development on, with a goal of being able to take the parameters and implement them across all of the isolators. So being able to actually do an initial distribution study and understand where the risk isolators were, if there were areas where we really didn't have to have biological indicators put on because they gave us repeatable inactivation profiles and a good efficacy. And as I mentioned, this limit testing, so understanding what those critical parameters are through the different phases of the gassing cycle, and really highlighting and picking up on all those challenges to efficacy, and making sure that the BIs would be able to be killed on those limit testing, so that when they're used in routine use, there's no surprises. There's not the risk that there could be a enzyme indicator that highlights that the cycle would have failed. So in terms of the outcome of the project, it was all successfully executed. Biological indicators and enzyme indicator results confirmed that there was robust repeatable cycles across all 8 isolators. And the strategy that was in place, and it was a well-documented strategy of how it would be achieved versus then how it was reported in the validation summary report, was all assessed as part of a regulatory assessment and inspection. What that fundamentally meant for the execution of the project was that the cycle development PQ, including that real robust testing of limits, allowed us to actually get 3 gassing cycles across 8 isolators in 6 weeks, versus if we had a traditional approach, which would be to take each isolator separately, that would have actually equated to 4 weeks for one isolator and waiting for results and waiting to move forward with confidence of the approach, which would equate to 32 weeks. So significant time saving, but so much more data that was gathered and be able to revert and work off that data. So that's all of the slides I have. Just to summarize, it's really about ensuring bio-decontamination efficacy is controlled and robust. And it is a challenging approach, especially as the feel and the thoughts have always been that you put the peroxide in, it kills the BI. That's not always the same approach. That doesn't always happen. There's a lot of things that can impact. So seeing the updated Annex 1 requirements is really about having that strategy. And with the enzyme indicators, it's about more data and more understanding of what that means to the approach. It's about having more process efficiency, having a tool that you can really understand, investigate. They're really a good question of the why. So if an enzyme indicator result is saying you've got a higher amount of relative light unit, why is it telling me that? What is it that is not right with the process? And it gives you that investigational tool. And it enables that process understanding, which is a must for Annex 1. And that significant reduction in cycle development and validation timelines also helps. I don't think many people would be given 32 weeks to execute their validation. And it's normally the last point of equipment being set up. So it's normally the part that gets squeezed on the program as well. And our goal as a company and how we're supporting our customers and how the focus group goals are is to really continue to share this information where we can, get that messaging out there, get that information, and really put that into best practice documents. So the results and data that's been gathered by the focus group is going to be put into a PDA publication, hopefully next year, early next year, and really increase that regulatory awareness of enzyme indicators as well. So that's all my slides. I'm just going to see if we've got any questions. Can't see any. Has anyone got any questions that they want to ask?
Oh yeah, it's much easier to ask, Kate, than it is to
Yeah.
type. So, Tim Triggs, I'm the chairman of the CCN. First of all, Kate, thank you. That was really clear and really well presented. And
Thank you.
I appreciate you sharing that with the CCN and some of our members today. Um, it's a topic that really interests me personally as well from my career history with GSK and aseptic and sterile manufacturing.
Mhm.
And so this holy grail of 6-log reduction has been part of our career for all time, I guess. And this shakes it up a little bit, I expect. I think what I was understanding was that this is something new on the block, which is shaking up the tradition of a 6-log reduction of a Bacillus stereothermophilus with a d-value of, and that's what
Mhm.
we've always done, and that's what we've always known. But I'm also sensing this is at the moment, maybe is this complementary to BI? So it's EI for process, BI for validation still. Is that the expectations of the regulators? Or do you sense that there's a shift in MHRA, FDA, and some of the international regulators to accept EI instead
Mhm.
of BI? How do
Mhm.
you feel that is shifting?
Yes, so there has been a huge shift. So when I started at Protac, it was very much a, is it approved by the regulators? And it was a very much almost like a closed door conversation. And biological indicators aren't approved as such by the regulators. They're just they're the industry standard for, they've almost been grandfathered into that approach. So it was really about using that data, educating on how they're a validation tool, using them alongside, and the feedback from the regulators have been really positive on that. So there's no preclusion to use in any of the guidance. And if you're using biological indicators, you're still using them. You're ticking the box that you're using biological indicators. But the guidance doesn't really tell you how many to use, doesn't tell you what your strategy is. So it's really having that robust strategy of use. FDA feedback we've had has been about having something that you can understand challenge locations. So how many times have you had to come up with where your challenge locations are? And it becomes, as much as people try to avoid it, it is a bit of a guess, and it's a bit of historical knowledge. And this is again trying to move it from having to rely on someone's knowledge of how they used to do validation and how they're using individual knowledge of how much gas to put in, to actually use something to give feedback. In turn, that means you can move away from biological indicators once you've performed the validation, because the goal of the validation, and you could only have 3 cycles really to show it's been validated. But how much data does that really give you to say it's robust and repeatable? Once you've done that, it's about then using enzyme indicators to remain in that state. So there's been a huge shift over the last 18 months in that on that approach. So
Yeah, thank you. I think that big win of what you shared in that study for NHS particularly was the time saving in cycle development, and to get you to a point of validation,
yeah.
you get much faster. Um, you One other question, if you allow me,
Yeah,
is
absolutely.
what your approach is to this as Protac. You supported the NHS study. It sounded like a project, a combined project
Yeah.
with Enver and others. I know there's a few people involved
Mhm.
at Royal Liverpool, but your role there. What is Protac's approach? Is it to you've developed a bioluminescence detector? I like photometry and bioluminescence, and that process is very robust. Is this an instrument now that's for sale to industry, so that
Mhm.
pharma, healthcare, life sciences would invest in this tool, or is this a service industry where you would support cycle development and assay? How do you see that going into
Yeah.
industry?
So
And you
that's a
never
really good
mentioned any money
question.
as well. No money to understand. But
Exactly.
given a kind of an idea of
Yeah.
save time, but how much does that cost as well?
Yeah. So it's a really interesting one, and it depends on what the project is. So there's always a depends, and there always is in
Sure.
validation.
Yeah, yeah.
We have models where it's a purchase, it's a training. The key part of our company is we are not a consumable seller. So we're a solution seller. So we don't sell the enzyme indicators and a reader and then just let someone come up with how they're going to use it. We coach, we mentor, we help them understand the data, because there's no benefit for us if that's the way the product's being used. And because there's so many differences in the peroxide bio-decontamination, that data, it has to be understood. And it takes that time to understand what it's telling you and how to use it to its benefits. So we have different scales of projects. We can be consultancy. My team are all ex-pharma validation experts in biodecontamination. So they have that experience to be able to look at different systems, understand how the gassing works. And that can be from doing a whole validation lifecycle package, or that could be going in to do an investigation. So we have real scalable projects, and it's all project dependent on that approach. But it will always be like that. I mean, okay, if we said 2, 3, 4 years time, our goal is that they're a lot more routine and being used a lot more routinely. But because the systems are so different and they give different results every time, there's always going to be that aspect of training and understanding. So
yeah, thank you very much. We're in Asia next week. You've done this a lot in the UK and in Europe. I like that you're going towards PDA. I guess this is a global solution that you're
absolutely.
promoting.
Yeah. So we are a global company, and we have large pharma companies in global rollout now. So it's a technology that, once it's understood and what it can bring, and that investigation piece. So some of the strategies are using them for information only on requalification, because if they know that they might have a failure, they've already gathered some information to give them the head start of where that issue is. And that's shifted a lot from a couple of years ago. We would have been, oh, we're getting a new isolator or a new setup, and we'll look into using enzyme indicators there. So that routine use is really the key strategic change and goal. So
no, thank you. I think it matches what we're trying to do in environmental monitoring of controlled environments with rapid microbiology
absolutely.
and the methods that are being promoted. So I really like that. I'll talk to you outside of this as to
yes,
whether
definitely.
we can mention it in Singapore next week.
Yeah.
Thank you very much.
Okay, thank you.
Could I just ask one question there? Excellent presentation, fantastic.
Thank you.
Just what are your thoughts on, you still have to verify the results using BI indicators, when BI indicators and the slide you put up almost require the 3 BIs at each position, where some MHRA inspectors would say it doesn't matter if you have 3, if one fails, it's still a failure. It seems like you're verifying a digital clock with a sundial.
Absolutely. And that is a challenge. And there's been, because of the variation in how the peroxide systems work and what we see against different solutions, and even now with all the different atomized, nebulized, ionized, there's so many different forms, and they're all the best ones. It's the being able to actually say, what's the consistency, what is that goal. That's all being worked on, all the data is being looked at, especially over the focus group, to come up with a common relative light unit that essentially means it's irrelevant what biological indicator you're using, what d-value. The issue is we're trying to pitch against something that can be a different d-value And then regardless of what the D value is, you may have a failure that doesn't mean your system's not working correctly. But it's to recognise, and it's an easier path in to say it's comparable or better information. The goal is absolutely that. So we'll canvass different systems. The data is all being generated at the moment to really look at what relative light unit value of inactivation do we get to that means that we no longer have biological indicator growth. And then once we get to that, that means it really makes it a lot of simpler. And then it could just be that you, so we have an OEM, for example, who is looking at setting all their cycles up, doing all their development, and significantly reducing when they actually include biological indicators. So it's very flexible in that approach. But
and do you have engagement from the regulator?
Yes,
in that respect.
Yes, it's very much we've had really good feedback on individual customer projects. So for us, they're not necessarily interested in regulating us or giving us the feedback. It's how the customers are using them. And the main thing is your strategy sound? So have you got a good, robust strategy? And that's really the support that we give to. It's not just about using them as a consumable item. How do you use them? How are you going to use the data? What's that investigation tool? So, and it's been really good feedback.
Yeah.
So
excellent. Thanks again for the presentations. Excellent.
Thank you.
Thank you.